Now let's look at Mendel's theory of segregation. Mendel proposed, it's his first theory, and that is that the organisms always have pairs of genes, and these pairs of genes are present in homologous chromosomes. And during meiosis, when genes separate, each gamete will go on one side of the cell, and resulting in the separation of the alleles. So for example, if we were to start doing a cross between these two combinations, that is capital A and small letter a, notice that the parental generation is capital A, and the offspring are and the other parental generation is small a. So if this is the parental generation will form two kinds of gametes. That is, one is going to be uppercase A and the second one is going to be uppercase A. However, the other parent, which has lowercase, will then result in alleles that is going to be lowercase a and a lowercase a. So now, in order to just look at the different combinations that are chosen for from these combination, Mendel designed a simple square that is called as a Punnett square. In a Punnett square, what he has done is he has crossed. For example, let me make you a Punnett square of this scenario. So if we were to do a Punnett square of this scenario, we will put in the two female or the male gametes on one side and the other two on the top side. So I put the uppercase A's on this side, and I've placed the two lowercase a's on this side. Now when I do a combination, I can see that my capital A can combine with a small a capital A with a small a. Now notice what is the appearance of each one of them. Each box is showing that it has a combination of capital A, and a small a. Now if capital A represents purple color flower, then all the offspring that are produced over here are going to result with the same appearance, that is purple. However, in the next stage of crossing, that is the next generation, which is called as now the second filial generation, notice that the A and the A will now separate out in alleles, and when it does that, you have a capital A, small a, capital A, small a. Now notice how they are written. You've got the small a and small a combination over here. You put in the capital A and a small a here. You do the same thing on the top lines, and then you do the same thing over here. Notice that the ones that I'm circling are the ones that are showing now as the homozygous gene. So you have one AA and one small a, and the other two that are shown over here that have capital A, small a, and there are two of them. So what has happened? The alleles have mixed, and in this case, the combination that I get is one dominant, two mixed, and one recessive, and this is called as 1:2:1 combination. However, because the one that has two and the one that is one have both the appearance of the dominant characteristic, the end result of this cross would be a ratio of 3:1. So this is the same ratio that is now shown on a next cross, in which we can see that the crosses between an F1 monohybrid will always result in the same typical ratio among the F2 offspring, and that ratio is always 3 dominant to 1 recessive. So this is the monohybrid cross, and the ratio is 3:1. Between the three, if I was to break this down, I can see one is a homozygous, and there are two heterozygous recessive. But because these two are dominant is the appearance, and this one is the homozygous, so altogether you have 9:3:1 is the ratio. I've seen students making mistakes when making gametes, so please make in mind that the parental generation you will always represent an individual with 2 alleles, and then make gametes by separating out the 2 alleles. Now, the second theory that Mendel proposed is called as the Mendel's theory of independent assortment. And in this law, Mendel proposed that the gene pair of the chromosome will independently sort and be present in the 4 different combinations of alleles. Now, this is because when the homologous chromosomes were aligning, if you recall the metaphase stage, when they were aligning on the spindle, if you recall this part, we said that the chromosomes were present in the center, in the equatorial region. Now, at this time, chromosomes can align randomly. So when they do that randomly, that will result in a simple experiment. We will look at a dihybrid cross in which we are looking at more than one assorted chromosome. So let's look at the dihybrid cross to understand this section better. Let's start with the cross between two true breeding heterozygous parents, and they're starting with a cross. Firstly, with this is a homozygous parent and this is a homozygous recessive parent. So first, let's say this is the dominant trait and this is the recessive trait. Now, with all the individuals that will form from this combination of AABB, we'll have the alleles would be capital A, capital B. You see that because that's the only combination possible. However, on the small one, you will have the combination of small a, small b. So now when I look at the dihybrid together, then in a dihybrid you will have 2 different characteristics, and it is showing you as now resulting in a heterozygous combination. Now let's look at the combination of alleles that are being formed after the heterozygous combination. The kinds of alleles that are shown over here is an example of heterozygous. Now, if you have capital A, small a, capital B, small b, first look at all the different kinds of genetic ratios that you can get. Alleles would be capital A can now combine with capital B. Capital A can combine with small b. Small a can combine with capital B, and small a can combine with small b. Now each of the combinations that I just showed you are the different alleles that are shown over here. So that's the same combination that's shown over here. Now, since we have a cross between the similar heterozygous, so we have the same over here. Now, when you do a cross in this pattern and you look at the ratio of each, you can see over here the combination. Notice the combination over here. You've got capital A, capital A, capital A, capital A, small, capital B, capital B. So look at all these combinations that are shown, the ones that I am highlighting over here. And these combinations will now result in 9 individuals that have dominant on both traits, 3 individuals that are dominant for A, 3 individuals that are dominant for B, and 1 that is recessive for both the traits. This is the combination that will always result when you have a dihybrid cross, 9:3:3:1.